So I'm going to talk to you about pluripotency. I should also say it's great to be back in the IMP. I spoke here in the past, I think 3 times, but not for quite some time. So it's very nice to be back in this splendid new building and auditorium. And yes, I'm going to talk about pluripotency. So this is the beginning, or kind of beginning. Of course, there had been work before this, but this was really the key experiment that set up our field. Very beautiful and simple experiment, the generation of embryonic stem cells from early mouse embryos. And many of the important breakthroughs in science, there are 3 key things that contributed, in my mind, to this breakthrough. Of course, there's an insight or some glimpse of understanding. And then there's perseverance and commitment to the idea. And then there's a sprinkling of good luck. Because as Meinrad's alluded to, what we didn't know then was that this experiment could never have worked if they hadn't, by chance, used one particular inbred strain of mice. But it worked, and it changed everything, and it gave us embryonic stem cells. So why are embryonic stem cells wonderful? So they're pluripotent. They can make all of the types of cell. The beauty of this is, this is not like cells in C. elegans or in Drosophila. There's no predetermination here. There's no pre-patterning. These cells are completely flexible in plastic. They'll go anywhere. It depends on the environment. In the embryo, this is completely controlled. In culture, we have a problem because we can't really control them so well, and this is one of the challenges. So this is a tabula rasa. Nothing is yet written in these cells. But the great thing about embryonic stem cells is this is also now an immortal property. So these cells can cycle forever and maintain exactly the same, if we culture them in the right way, exactly the same quality of pluripotency. And so then we can play with them and do whatever kind of manipulations we're clever enough to think of. So how does this relate to pluripotency in the embryo? So pluripotency in the mammalian embryo lasts a few days in early development. So pluripotency is generated in the blastocyst within the inner cell mass. But this population of pluripotent cells changes dynamically over the next couple of days in the mouse embryo. And pluripotency goes on for a further day until the end of gastrulation. So this initial apolar ball of cells becomes organized into an epithelial layer, undergoes this morphogenetic process. And then these cells start to become regionalized and patterned and really primed for gastrulation. And so during this process, we see a complete switch in the key transcription factors and signaling pathways. We see the emergence of specification pathways. We see changes in the epigenome. We see a dramatic increase in DNA methylation globally. We see an increase in polycomb activity. We see a change in metabolism. So this pluripotent cell population is changing as much as you would see in any differentiation event. And yet, these cells at the end, although they're already fated, we now know exactly what every single cell in the embryo is going to become at this stage. They are still pluripotent because if we move them around, they can change that fate. But this is a different kind of pluripotency from the cells here, in which nothing has yet been written. Nothing has to change for them to be able to do everything. So, there are many questions we can ask about pluripotency. I'm going to touch on the 3 questions that I think are the most important today. I'm really only going to talk in detail about one of them, or about part of one of them. But I'll just touch on this one first, which is what really is the relationship between these self-renewing embryonic stem cells and this process of dynamic, continuous progression that we see in the embryo, where really it's highly debatable whether there's any self-renewal, because the cells are changing all the time. So what we know is we can't make embryonic stem cells from all these stages of pluripotency. We can only get them, real embryonic stem cells, prior to implantation, and actually from a very finite window of less than 24 hours. And this is immunostaining for OCT4 in red, which is staining all of the inner cell mass, and then in yellow for NANOG. And NANOG is specifically staining this epiblast compartment. And these are the cells that will give rise to ES cells if we put them in culture at this moment in the right conditions. And these cells will only stay in this state in the embryo, actually for about 12 hours. But once we put them in culture, they'll stay there forever if we keep them in the right condition. And we know that globally, at the transcriptome and the epigenome level, they are very, very similar to this state in the embryo. But they reveal this infinite propensity for proliferation. So it used to be considered that embryonic stem cells were very homogeneous populations, and that pluripotency was a property of every cell equivalently in the culture. But after we had found this gene NANOG, that we considered to be a central element of the gene regulatory network, and which I've shown you is uniformly expressed in the early embryo, and is actually essential for formation of the epiblast in the embryo. And we were really shocked when Ian Chambers, who discovered NANOG, performed this immunostaining, expecting all of the ES cells to be NANOG positive, just as they're OCT4 positive. And this is what he saw, that they show this range of expression. And if you make a fluorescent reporter, you can see that the cells are switching NANOG on and off. And it turns out this is true of several transcription factors in ES cells. So this is another one, REX1, hooked up to a GFP reporter. I'm going to use this reporter quite a lot in the talk. And what you see in ES cells cultured in serum or in LIF and BMP, you see this bimodal distribution, cells that are positive and cells that are negative. And the positive cells will give rise to the negative cells, and then occasionally the negative cells can become positive again. So this kind of dynamic heterogeneity caused a great deal of heat, excitement, and people thinking, well, this sort of metastable situation reflects coexistence of different states, and that's what pluripotency and stem cellness is all about. And I even wrote a review putting forward such a possibility, which I'm now a bit embarrassed about because I think it's completely wrong. That is not at all how it is. It's an artifact of growing the cells in a very messy culture condition and giving them conflicting signals. And the reason I think that's the case is because this is not how development works. Development doesn't go backwards and forwards. Development goes in one way, one direction. So, development, as I showed you, we have this progression, this ever-changing pluripotent population. And this is how we are currently thinking of this landscape of pluripotency. That cells, pluripotency initially emerges in this naive, erased, blank slate state that has to be created early in mammalian embryogenesis. This is very different from the zygote that is still maternally programmed. But this cell state, we hypothesize, is not at that point competent to generate any differentiated progeny, but has to go through some process to become capacitated, to gain competence. So we call this phase the formative phase of pluripotency that corresponds to this peri and immediate post-implantation period where the cells have changed a lot but haven't yet gained any regionalization or specification. But then they're in a stage where they can respond to specification cues and become primed or fated, but still not committed. But from that point then, these cells are poised to enter into gastrulation. And what we think is happening with this epiphenomenon of heterogeneity of ES cells in serum or in LIF and BMP is that they can wander around in a deregulated way in this landscape. But that's not how ES cells have to be in culture. Because if we culture them in a defined condition, then we capture them exactly, as far as we can see, corresponding to this population of cells in the early embryo, and all of the cells are in or very close to the same state, which is why we call this a ground state, because the cells are homogeneous. And this is showing you this. So this is, as Meinrad alluded to, using two small molecules to inhibit two signaling kinases, ERK-MAP kinase and GSK3. And in particular, if we combine that with LIF, then what you see, in contrast to what I showed you before, all of the cells are nanopositive, all of the cells are REX-GFP positive, all of the cells are positive for all of the key transcription factors. So it's a very homogeneous population. Then, of course, people say, well, this is obviously an artifact because you're using small molecules. Well, I'll say it looks like an artifact if you can only make ES cells from one inbred strain of mice. Now, I can make ES cells from any strain of mice, just from a single cell from an embryo, and also from rats, another different species. And the important point here, a really powerful thing about mouse ES cells is you always can show you've really got an ES cell. There's only one thing that matters, which is, can it make an animal? Can it make a chimera? And these cells can do this, and of course they can go through the germline. So, although we may be using small molecules, what we believe we're doing here is capturing something authentic. And so with that in hand, we're able then really to delineate and to dissect the core gene regulatory network. And so these are the factors, the transcription factors that we believe, based on our work and work of many others in the field, are key to the naive ES cell state, mouse ES cells. So we have the two central factors, OCT4 and SOX2, that all kinds of pluripotent cell require, but that are not specific to the ES cell state or just to this early naive epiblast. But these outer group of factors are all specific to the ES cell state and to this pre-implantation epiblast. And they are regulated by the two inhibitors and by LIF. So the only place you will find all of these factors together is at this moment that I showed you on the earlier slide of less than 24 hours in the pre-implantation mouse embryo and in ES cells. And these factors are all cross-regulating one another, and you'll generally find them co-occupying the same target genes. So there's a very high interconnectivity in this network, and there's a redundancy. So of these outer factors, you can remove any one of them and you'll still have pluripotent ES cells. They could be more or less destabilized, but they can still maintain pluripotency because in the mouse this network is so resilient. But you can never remove OCT4 or SOX2. So if we have such a resilient network, well, okay, actually, there's one other thing that is important, I think, to point out about ES cells and this idea of a tabula rasa. That ES cells cultured in these ground state conditions they are completely independent of DNA methylation. They don't care about it at all. They don't really care about histone modifications, about polycomb activity. So this is really quite unusual for mammalian cells. So they're really not using the conventional epigenetic machinery to program their identity or maintain their identity. But cells with mutations in these pathways are always compromised when they begin to differentiate. So this epigenetic machinery becomes important for the cells progressing from this naive state. And this leads us to what I want to talk about now, is this idea then that there is some key process of capacitation, which partly involves activation of epigenetic machinery, that renders this naive state ready and responsive to lineage inductive cues. Because the naive state is really momentarily created as just a wiping the slate clean, as a starting point, where you get rid of everything, but then actually you have to start to rebuild things. So we've created this robust network to get rid of everything, but then how do naive cells develop this competence for lineage formation? So how do they get out of this state and change into something else? So in the mouse ES cell system, this actually happens really pretty fast. So when you withdraw ES cells from the 2 inhibitors, 24 hours later, you can see already they've dramatically changed in morphology. They've already begun to adopt this more epithelial, flattened morphology. We see downregulation of the naive factors with somewhat different kinetics, but OCT4 here is not really changing. And we see, with similar kinetics, the upregulation of new transcription factors and signaling molecules. And if we take these cells at the 24-hour time point and we sort out the REX-GFP nanog positive cells from the REX-GFP negative nanog negative cells, and if we take that negative population of cells, they have lost the ES cell identity. So if we put them back in 2 ion lift, they can't make colonies anymore. Right? So what can they do? What we find when we take that population, which in blue here, these are the negatives, the reds are the positives, then what we see is that they respond very rapidly to inductive cues. Here for primitive streak-like cells that are brachyury positive, here for neurectoderm cells that are SOX positive. So they're responding almost immediately to these cues. So faster than the positive cells, which are still essentially ES cells. Perhaps most persuasively, these REX negative cells are now able to make germ cells. So of course ES cells can make germ cells. But actually, if you start from a naive ES cell, that cell is not able to respond either to cytokines or even to transcription factors and directly become a primordial germ cell-like cell. So that is the work of Mitanori Saito and Katsuhiko Hayashi and Azim Sarani. And what they have shown quite clearly is that the ES cells have to be transitioned from 2I for between 24 and 48 hours. In their system, they treat the cells with FGF and activin and KSR. And then, and only then, can they become responsive to these signals to acquire the germ cell fate. And what we show here is that actually, if you sort the REX negative cells at 24 hours, in a condition where they haven't received any cue at all, then they've gained this competence. So what is going on here? Again, if you read the literature, what you'll see, what everybody talks about is switching from naive to primed states. But if we look at the gene expression profile here, then these are our starting ES cells on the left, and on the right are the REX negative, nanog negative cells. And what we see here is that there's no change in expression of genes like OCT4 and SOX2, the core pluripotency factors, but the naive factors, this outer group of factors like nanog, KLFs, they're the genes that are being downregulated. But if we look at the lineage specification markers, genes like T, FOXA2, SOX17, Cerberus, those kinds of genes that come on in the later epiblast, none of them are upregulated at all at this point. So there's none of what people would consider to be priming in terms of early expression of these lineage-affiliated genes. So what is upregulated here is a relatively small group of genes, including the de novo methyltransferases, which do play an important role here because there is a big change in DNA methylation in this population. But I'm not going to go into that today. I'm going to highlight 3 genes: PAL3F1, also known as OX6, LEF1, and also OTX2. These are 3 transcription factors that we believe are pivotal in this next phase of pluripotency. And what you can see is that they actually begin to be upregulated from 16 hours before there's been any loss of ES cell identity. And this is mirroring what happens in the embryo. So this is a very nice study from Antonio Simeone's lab in Naples. And they work a lot on OTX2, and here they're looking at OTX2 and also OX6. So this is similar to what I showed you earlier, that in the mature blastocyst, then the inner part of the inner cell mass, the epiblast cells, are all nanog positive, right? And the OTX2 low or negative at that point. So interesting, the OTX2 is expressed earlier, then it goes away as nanog comes up, as the naive epiblast is born. But then, as this embryo develops to the point of implantation, nanog is downregulated, it goes away, and OTX2 comes up. And so you end up with a population at the time of implantation that is uniformly OTX2 positive and nanog negative. And OX6 appears at the same time. So just as we see in the in vitro transition. So this is happening very fast in the mouse embryo. Just to summarize this part, the cells that have newly departed, the first cells that we can isolate that have left the ES cell state, because they can no longer make an ES cell colony if we put them back into 2-ion life. And this is driven purely by the signals they're making themselves. In particular FGF4, but we also have some data indicating that nodal is important for the future capacity of these cells. So these cells have gained responsiveness to germline and somatic lineage induction, but they're not expressing any regionalization or specification markers. So to my mind, it makes no sense to call these cells primed. That's what will happen in the next 24 hours when they receive specific cues. This then is what has made us propose this idea of a formative phase of pluripotency that has been, I think, overlooked in the mouse, both in development and in the ES cell system. Because it's so quick, it's actually a very difficult stage of mouse embryogenesis to study this period immediately after implantation. And in the mouse ES cell system, not only is it fast, but because the cells are behaving asynchronously, unless you do single cell studies or have a reporter like RexGFP, then you always see what looks like mixed gene expression. But actually things are behaving in quite an orderly progression. And what we believe is going on here is that there's a complete switch in the gene regulatory network. The naive factors disappear completely. There's a major chromatin reorganization. Most obviously it's seen at the level of global DNA methylation. There's enhancer rewiring. There's, again, an early and dramatic change in metabolism, loss of mitochondrial activity. The cells become polarized and epithelialized, and we see altered responsiveness to signaling pathways. And associated with this is the decommissioning, the loss of the ability to make extra embryonic lineages, trophoblast and primitive endoderm. So collectively, these events lead to this generation of germline and somatic competence. And in a way, this is the business part of pluripotency. That if we're really to understand what pluripotency is, we need to understand these events and how they come together to create this competence. So just to illustrate this in another way, human naive pluripotent stem cells, which can now be isolated both from embryos or by reprogramming somatic cells or by converting established cells, conventional human pluripotent cells. So these cells, although it's still contested to some extent about exactly what their identity is, they're clearly pretty similar to the human naive epiblast. One of the issues that has concerned people about these cells is that they don't immediately differentiate when you put them into the protocols that people have developed for conventional human pluripotent stem cells that represent a later stage of actually gastrulation stage epiblast as far as we can see. They don't differentiate into protocols that are actually reasonably efficient for conventional cells for generating neuroectoderm, dual SMAD inhibition, or for generating definitive endoderm, or actually the same is true for mesoderm. But if you remove the cells from the self-renewal factors, which is essentially a variant of 2-iron LIF, human cells, for 6 or 7 days, then what you see is that they gain a high level of responsiveness to exactly these same inductive conditions. So this process of capacitation that we're proposing then is taking a lot longer in human and mouse. And so then people say, well, you know, something's wrong. Why should it take so long? And my answer to that is actually you're asking the wrong question. The question is, why is it so fast in the mouse? Because if we look at development, indeed, in the mouse, this process is very fast. It's 24 hours, this change. But if we look in the human, then there is a long interval between the naive epiblast and the start of gastrulation. So it's several days, 6 or 7 days. We don't know exactly in a human embryo. But what we also know is that this mode of development is the predominant mode of development in mammalian embryos, that rodents are exceptional in having such a truncated period before gastrulation because they're reproducing very rapidly, rodents. How is the rapid transition executed in the mouse? We have to study the mouse first because that's what we have the most information on and where we have the most robust cultures, and also it's where we have the embryo reference material that we don't really have in human. So how do the mouse ES cells make this switch? So over the years, we have carried out a series of, and in collaboration, a series of genetic screens to identify players that may be involved in this process. And the principle here is very simple. We use our REX-GFP cells, we withdraw them from 2-iron LIF, and we do some genetic perturbation, and then we ask, is there any cell that resists loss of ES cell identity that we can identify by REX-GFP, and then what's the genetic defect in that cell? So the first gene we identified in this way was TCF3, also known as TCF701. And this gene is recovered in every screen. It's always the most powerful hit, has the most recurrent hit, always has the strongest phenotype. And indeed, in parallel work, we and others show that the TCF3 is really the key target of GSK3 inhibition in ES cells. So TCF3 acts as a repressor, and it shuts down key members of this outer group of factors, in particular ESRRB and ANOG. So we then performed several other screens, RNAi screens, haploid screens, and identified quite a large number of genes, all told. So what can we learn from this? So many of these genes are involved in shutting down the naive circuitry. specifically this outer group of factors, and ensuring that there's a rapid and complete dissolution of this program. So these factors are targeted at multiple levels, at transcription, at protein stability, at protein localization. So the circuitry is completely disabled within 24 hours. So there are many ancillary factors that are supporting TCF3. Nothing has as strong a phenotype as TCF3, but many other factors have some phenotype. And these include components of regulatory pathways, of course the ERK pathway, TORC and AMPK that regulate folliculin and TFE3, protein kinase C iota, that in fact turns out to be a key signal to inhibit in the human system. There are some factors that are specific for pluripotent cells. And then interestingly, there are a bunch of factors that I would classify as sort of generic tuners, fine tuners of gene expression. So it's not really that they're doing anything very specific to the pluripotency circuitry. But as cells make a transition, and as gene expression may become more noisy, and it's more important to keep it within critical bounds, then these factors, I think this is most clearly shown by Brian Hendrick's work on the NERD complex, act to keep the gene expression relatively orderly so that the cells can prosecute a transition. But the key point I want to make is that none of these factors blocks the ES cell transition. So the cells in the end, even without TCF3, they will eventually escape the ES cell state, and then they're able to differentiate completely normally. So we then became interested in, although most of the genes we're pulling out of the screen are driving collapse of the naive network, that can't be enough for the transition, right? The cells have to transition to something, and that something has to be set up properly. So how is this new network initiated? And we were particularly interested in the idea that the ERK signal, although it's playing some role in dissolution, should be activating this new network. So we went back and looked at the screen hits, and in particular for genes that might be targets of ERK and that might be activators. And this gene, ETV5, stood out. So this is a recurrent hit. The phenotype is quite weak, but it is a recurrent hit, and it's an ERK target. So ETV5 is an ETS domain protein, so these are a family of transcriptional activators that are transcriptionally induced by ERK and are also phosphorylated and activated by ERK. So when we look at ERK targets in ES cells, we find some interesting genes, including OX6, LEF1. I could show you exactly the same thing. So these are CHIP targets that are upregulated early during the transition, so not expressed in ES cells, upregulated, and this upregulation is substantially reduced in the absence of ETV5. That leads to the simple model then that ETV5 downstream of FGF4 and ERK is activating the succeeding network, specifically these two components, OX6 and LEF1. So if that's the case, then we might think, well, if we think that ETV5 is the key target of the ERK pathway and therefore of MEK inhibition in the 2I system for maintaining ES cells, and TCF3 is the key target of the GSK inhibition, then can we reproduce the effect of the two inhibitors just by deleting both genes, by combining a TCF3 mutant with an ETV5 mutant? This is the experiment here. We're looking at REX1, but we see exactly the same result if we then do colony assays on these populations. So the point here is that after 72 hours, the REX positive population has disappeared completely in control cells. They're all REX negative. TCF3 cells have a strong phenotype, so there are many GFP positive cells. ETV5 cells mutant cells have a weak phenotype, so there's just a shoulder of positive cells. But the double mutants actually look unchanged from cells in 2I. After 72 hours, it looks perfect. These cells haven't moved at all, or barely. But when you passage them, what you then see is that a prominent peak of REX-negative cells appears. And if you passage them again, their whole culture collapses. So this is not equivalent, cannot reproduce the effect of the two inhibitors. So we went back and looked for the possibility there might be some other key player in this system. And here we were struck by RBPJ. So RBPJ had been identified and validated in Martin Lieb's screen as having a strong phenotype, almost as strong as TCF3. But RBPJ, which is a repressor, transcriptional repressor, most widely known as part of the Notch pathway. But I want to say that there is no evidence, we cannot find any evidence that Notch pathway components are expressed or that the Notch pathway is active at this stage of the pluripotency spectrum. So we don't think this is to do with Notch, that it is reflecting a pure transcriptional repressor activity of RBPJ. And RBPJ has no known role in pluripotency. So we were interested then in could RBPJ be a key player here? And the other reason is that RBPJ is upregulated both in ES cells and in the embryo during this transition, both at the transcript level and actually we see a similar thing at the protein level. And that this upregulation requires the withdrawal both of the MEK inhibitor and of the GSK3 inhibitor. We made a triple mutant. Any double mutants have the same phenotype as the one I just showed you, that eventually the cells escape. But when we make the triple mutant, then the cells can be propagated long-term, so without any 2I, any LIF, just in a simple defined medium. And they remain almost entirely REX-GFP positive, just as if they had been cultured in 2I and LIF. So this is just showing data from two independent triple knockout clones. And they retain expression not just of REX-GFP, but they retain full expression of other naive factors, NANOG, KLF2, ESRRB. Similarly at the protein level, TFCP201, NANOG. The cells are a bit more flattened, so the morphology is a bit different, but the marker profile looks consistent with an ES cell identity. And they fail to upregulate these formative markers, FGF5, OX6, OTX2, without any exogenous inductive signal. Then these cells remain as ES cells. What happens if we try to make them differentiate? So if we aggregate them and make embryoid bodies, they fail to differentiate. We see some endoderm markers which may well be primitive endoderm, but we don't see mesoderm or neurectoderm. If we put them in FGF and activin, which is the standard condition people now use to drive ES cells into this primed epistem cell state, then initially they don't change at all, but after a few passages you do begin to see some REX-negative cells, but you can still maintain REX-GFP positive cells for many passages in this strong inductive regime. And when you characterize this population, so it's the bars at the end are the triple knockout cells, the ones in between are the different single mutants, and then these are the ES cells at the start. So of course they retain, all of them retain OCT4. The triple mutants express high levels of KLF2 and TFCP201, naive markers. They fail to upregulate, as I've already shown you, FGF5, but this is now in the context of not just a few days, after many passages. Upregulate FGF5, OTX2 or OX6, although the other single mutants do upregulate these markers because they can eventually transition. Actually, the ETV5 mutants can't, but the other two mutants can. So these cells then really can't progress in differentiation, these triple knockout mutant cells. So what's changed about them? Globally at the transcriptome level, they look pretty similar to ES cells in 2-iron-LIF, closer to those ES cells than even to a 16-hour withdrawn population. And what we see is that they have upregulated two of the ID genes, ID3 and ID4. So ID3 is normally not really expressed either in ES cells or in differentiating cells. ID4 is expressed in ES cells but downregulated during the transition, but in the triple knockout cells and also in the RBPJ single mutant cells, then these genes are misexpressed. And why is that interesting? Because a long time ago in the lab, before we had the two inhibitor system, Ying-Chi Long showed that you could maintain ES cells with LIF and BMP, and that the major way that BMP works is by inducing through SMADs the ID genes. So ID genes then are antagonizing escape of ES cells from the naive state. RBPJ is going to be opposing this by shutting down the IDs. So what we surmise from this then is that RBPJ enforces transition by suppressing this resistance mechanism of BMP, which we know is expressed in these cells as they're starting to differentiate, and also in the implanting embryo. And RBPJ, increasing expression of RBPJ, will be shutting down this effect by clamping the ID expression. So, I told you that the defining feature of ES cells is not which genes they express in the culture dish or how they look, it's how they behave in the mouse embryo. So if I want to argue that these triple knockout cells are still ES cells or somewhere in the ES cell state space, they should be able to make some kind of chimera. But of course they're also blocked in differentiation. So what's going to happen when we put them into the embryo? So we injected, this experiment was carried out by Tim Lohoff, a PhD student, with our collaborator Jenny Nichols. Well, actually, Jenny did the injections and Tim did the imaging. So Jenny injected cells into 8-cell embryos. And when you do this with good ES cells, they'll more or less take over the entire inner cell mass. So that the vast majority of cells in the inner cell mass, marked here by SOX2, in the epiblast specifically, so the GATA4 is marking the primitive endoderm. So the epiblast is, the ES cells have largely taken this over. And we have a reporter in our ES cells, so we can see that most of these cells are derived from the ES cells. And they've specifically gone to the epiblast. They don't go to the wrong places, primitive endoderm or trophoblast. What about the mutant cells? And this, Jenny has shown very clearly that this is very specific. So if you take Rex-negative cells, they will get eliminated with high efficiency from the embryo. And Hitoshi Niwa previously showed that. So as soon as cells have exited the ES cell state, they're not able to do this. The embryo recognizes they're wrong, or they recognize they're wrong, and they get booted out. And epistem cells cannot do this. So it's actually a pretty reasonable functional test for ES cell identity. When we take our triple knockout cells, they behave exactly as the parental cells. So they extensively colonize the naive epiblast, and they don't go into the primitive endoderm or the trophoblast. So at this point, they're looking just like normal ES cells in a 4.5-day embryo. But then we expect, based on the in vitro phenotype, that things should go wrong, or may go wrong, because they shouldn't be able to progress. They should be trapped in this stage, really. So what happens when we look post-implantation? So here's the controls. This is an early gastrulating embryo, shown here with brachyuria expression. So the Rex reporter now is completely downregulated. So we're using Rex rather than SOX2 here. So this is the Rex GFP. It's completely downregulated, of course, after implantation. The cells have gone everywhere throughout the epiblast, only the epiblast. very nice typical representative ES cell chimera. If we take our mutant cells, then what we see, in some cases the embryo is completely retarded and disrupted. In other cases we have things that look somewhat normal, but what we see is that the ES cell progeny have retained the expression of REX, so they've retained a naive marker expression, and they show this very abnormal distribution pattern of colonization and this extension into extra embryonic regions as though they're being extruded from the embryo. So, to summarize then, these triple knockout cells are stalled at some very early point in the transition, in which they can self-renew independent of the microenvironment, so now we don't need to block ERK, we don't need to give them lift. They retain the core gene regulatory network features of the naive ES cells, so NANOG, ESR, or B, etc. And functionally, they have that identity that enables them to integrate into the pre-implantation epiblast, but then they'll fail specification in the post-implantation epiblast. And if they're the major population in the embryo at that stage, the whole embryo will be disrupted and retarded. So, what's the point of all of this? So what we're trying to get at here is how the cells make a state transition. And we're looking at this very specifically at this first transition of ES cells. But you know, it's possible that there may be some general principles here about how cells transition. And we think that these three factors are at the top of hierarchies, and that may be telling us something about three sort of complementary activities that have to happen during such a transition. So there's a dissolution of the existing gene regulatory network that is driven by TCF3, but it has several friends to help it in this process. And then there's the activation of the successor GRN, which is being driven by ETB5, but probably has other factors. Now I want to make a point here, because again, you'll read in the ES cell, in the stem cell field generally, that what happens in these state transitions is that you have these mutually repressing factors that coexist, and then somehow one program becomes dominant. We don't see any evidence of that at all. These factors, this is a new set of factors that's being induced, and this happens really in sequence. These guys disappear, and new factors like OX6 and LEF1 and OTX2 come up. Now indeed, the two networks may co-repress one another to make sure that things don't go backwards. But there is really a sequence of events here. And we have single cell data that consistent with that. And then we see this third element, the RBPJ, which is something that we couldn't really have predicted, and I'd say we're still really trying to understand fully what this means. But what it looks like is that RBPJ is really enforcing the directionality of progression, and it may be playing some role in controlling the flow, the rate of at which ES cells transition, and making sure that this is irreversible by shutting down machinery that would otherwise delay the process and make it possible for the cells to go backwards, the way you see in a serum or a BMP LIF culture condition. So, to get back to the question I posed earlier then, why is the mouse faster than the human? Because I'm trying to suggest that this principle might be generic and it should be the same possibly for all cell state transitions. So how can we alter the timing? Actually, it's very simple, I think. You just change one or two of the components. And in particular, human naive cells completely lack TCF3. They also lack ETB5. And TCF3 isn't even upregulated until quite late. So what we think may have happened here is that during evolution, TCF3, which is normally part of the Wnt beta-catenin signaling pathway, has been co-opted specifically as a repressor to give this accelerated transition in rodents. And you don't see that in primates. It's not just in human, also in data from marmoset and from cynomolgus. TCF3 isn't there. And without TCF3, well, we already know that from the mouse. If you take TCF3 away, then things go a lot slower. Okay, so that's almost the end. This is my wonderful lab. What I've talked about today is almost entirely at least the latter part of the triple knockout story, the work of Tusa Kalkan, postdoc in the lab. And I'll just leave you with one last issue in our field. So Richard Feynman said, "What I cannot create, I do not understand." I think that's an important issue for scientists. It's what I always say to developmental biologists. You can describe it in as much detail as you like, but actually make the embryo. It's the power of ES cells. In principle, we should be able to do that. But, I have a problem with that as well. So now I can make ES cells from a mouse or a rat, but I can't make a real ES cell so far from anything else, including species that will be very interesting and useful to have ES cells from. And, some of you will be sitting there thinking, oh, well, we read all the time about people saying they've made iPS cells and ES cells from these different species. So I'll just remind you, an ES cell means only one thing. It must be able to colonize the entire embryo and to give a live bull-born chimeric animal. If it can't do that, it's some other culture thing. It could be a complete artifact, or it may be related to some later stage of pluripotency. And the point here is, only by doing this, what we're trying to understand, is there really some unifying regulatory principle to pluripotency? Early development is quite different between different mammals. But at its core, we think pluripotency, naive pluripotency, ought to be fundamentally similar, even though it appears to be different at this point. So I'll leave it there. Thank you very much for listening.